You have probably heard that humans are 99% chimpanzee. I am going to ruin that number for you in the next few minutes, and honestly, you will be smarter for it. Today, on Guess What I Found Out, we are digging into how similar humans and chimpanzees actually are, using genetic data so new it only came out this year. Stick around, because that famous 99% number can legitimately drop as low as 86%, depending on how you count, and neither number is lying to you. Let's call this whole mess the shape-shifting 99%, because that is exactly what it is, a number that changes shape depending on what you're actually measuring. People have known humans belong with the other apes for centuries, they just did not know why yet. Back in the 1600s, an anatomist named Edward Tyson dissected a chimpanzee and noticed its brain and body looked more like ours than like a monkey's, which apparently made a lot of people in Western Europe deeply uncomfortable. In the 1700s, Carl Linnaeus built the classification system we still use today: kingdom, phylum, class, order, family, genus, species. And he grouped humans with orangutans, chimps, gorillas, bonobos and gibbons because the physical similarities were impossible to ignore. But this was before evolution was even a scientific idea, so nobody assumed similar meant related. Species were just thought to be built from a shared blueprint, full stop, no family tree required. That changed in the 1800s with Darwin and Wallace's theory of evolution, and later with the discovery of DNA and genetics in the early 1900s. Suddenly, similarity had a mechanism behind it. Related organisms share genetic material because they share ancestors the same way you share more DNA with your sibling than with a stranger on the street. In 1975, researchers Mary Claire King and Alan Wilson compared a batch of proteins between humans and chimpanzees and found they were about 99% identical. That number stuck in the public imagination and has been repeated in textbooks and documentaries ever since, usually with zero explanation of what it actually measured. Early subscribe nudge here, because if you like watching a tidy scientific fact slowly fall apart under closer inspection, you are exactly who this channel was built for. Here is the problem. Your entire genome is made up of billions of DNA letters, but only about 2% of it actually codes for proteins, the working parts of the cell that build and run your body. That tiny slice tends to be extremely similar across related species, because it does these critical, non-negotiable jobs, so evolution keeps it locked down tight. Too much change there, and the organism usually does not survive. The other 98% is regulatory, telling genes when to switch on or off, or it is just repetitive filler along for the ride. And honestly, it is where most of the real differences between species actually hide. When scientists finally sequenced full genomes for humans and chimps in the early 2000s, they found that once you include small, single-letter mutations across the entire genome instead of just the protein-coding slice, humans and chimps were closer to 96% similar, not 99%. But here is where it gets messier. Whole genomes are not just made of single-letter changes. Big chunks of DNA get deleted, duplicated, or flipped backward entirely. And those events can shift enormous stretches of sequence out of alignment, even though only one mutation actually happened. Picture copying a paragraph and pasting it in twice by accident, or flipping a whole sentence backward. Technically, that is one small edit, but a computer comparing the two versions, letter by letter, would flag the entire duplicated or reverse section as different. Genomes do this constantly. When researchers tried to line up two full genomes and count every unmatched piece as a difference, the similarity score dropped dramatically, in some calculations down into the 70s or 80% range. And here is the part that will really mess with you. When scientists ran that exact same comparison between two different human genomes, the number was not 99.9% either. It landed around 92 to 93%, purely because of how repetitive, jumbled regions of DNA behave, even within our own species. Same species, same comparison method, wildly different number than the one everybody quotes. Quick comment prompt, because I want to know where you land on this. If protein coding regions say 99%, whole genome mutation counting says 96%, and raw alignment says somewhere in the 80s, which number do you think actually deserves to be called the real answer? Drop your reasoning in the comments. I read every single one. This year, researchers finally sequenced complete gap-free genomes for 6 ape species: chimpanzee, bonobo, gorilla, 2 species of orangutan, and the siamang, using new long-read technology capable of reading straight through the repetitive junk that used to be, well, pretty much unreadable. It took over 100 scientists across more than 40 labs, and honestly, this is the first time we have ever compared full ape genomes without any missing pieces at all, including those notoriously messy regions like the immune system genes that help your body recognize threats. And you know, the new timeline it produced has some real surprises. Humans and chimpanzees split from a common ancestor between about 5.5 and 6.3 million years ago, which lines up closely with older estimates. Gorillas split off a bit earlier, around 10.6 to 10.9 million years ago. But orangutans split from the rest of the great apes much earlier than we thought, somewhere between 18 and almost 20 million years ago, which is nearly double the human-chimp split. The same paper also flagged specific genes that appear to be uniquely human, several of them tied to how our frontal cortex develops, which is about as close as genetics gets to answering what actually makes us different, buried inside that shrinking but still meaningful gap. If you liked watching an entire wolf population blow past what scientists expected of them, go check out our sea wolves episode, because, honestly, biology just does not seem to care about the tidy categories we build for it. Whether that is a wolf eating sea otters or a genome refusing to sit still at one clean percentage. This same messy comparison problem shows up whenever we compare any two genomes, by the way, not just humans and chimps. It is basically the same logic we use comparing dogs to wolves, which we covered back in the Firelight Freeloaders episode. Dogs and wolves are each other's closest relatives, no matter which counting method you use, even though the exact percentage wobbles depending on the math. So, how similar are humans and chimpanzees, really? In protein coding regions, over 99%. Counting small mutations across the whole genome, about 96%. Counting every structural difference as a difference, somewhere around 86 to 90%. All 3 numbers are technically correct. They are just answering different questions. What does not change under any method, in any paper, using any technology, is that chimpanzees are still our closest living relatives. And frankly, humans are more similar to chimps than tigers are to house cats, than Asian elephants are to African elephants, or than rats are to mice. So next time you see a headline screaming that humans are 99% chimp, or worse, that we are barely different from chimps at all, you will know there is a lot more going on underneath that one tidy number. Numbers do not lie, but they absolutely can be cherry-picked, stripped of context, and turned into a clickbait title. And now you know enough to catch it happening. That is honestly the whole point of a channel like this one, not just handing you a fun fact, but handing you the tools to question the fun fact the next time somebody hands you one. Hit like, subscribe, and let me know in the comments which similarity number you think should get top billing. Thanks for watching, and I will catch you in the next one. See you there, Curious George.